right, in this video we are going to wrap up our chapter on kind of the overview of the nervous system and neuron physiology. All right, so we are going to go through now neuron physiology. So in a single neuron, how do they receive information and process that information and send it down their axons to communicate with other neurons and that sort of thing. Okay, so with neurons, all right, we can have either continuous conduction, or we talked about last time saltatory conduction. All right, so saltatory conduction would be if these are our myelin beads, right, that information would jump from one node of Ranvier to the next. All right, so that's saltatory conduction. Continuous conduction would just refer to it moving down an unmyelinated axon. Okay, whoops. All right, so this would be for an unmyelinated axon. All right, and this process, because it has to move down without jumping, right, from node to node, is much slower than saltatory conduction. All right, and so we are going to look at primarily saltatory conduction, right, because we're going to be focusing on myelinated axons. Okay, so with this process, we are going to start in our dendrites. All right, so we talked about last time our EPSPs and our IPSPs, okay? And so we're going to start in the dendrites, and it's important to point out with this kind of picture we're looking at, this neuron, the main neuron we're looking at, all right, here, is called the presynaptic neuron, all right? And that's because, so presynaptic, all right, because this neuron is the one that is sending the information, okay? So it's going to receive information from another neuron, right, process it, and it's going to send that information. Okay, and it's sending it to the postsynaptic neuron, right? So the postsynaptic neuron is always going to be the one receiving information. Okay, and so that's just referring to the synapse we have here, right? So this would be a synapse, right? That kind of space where one neuron meets the next, all right? So the presynaptic neuron sends the information towards that synapse, all right? And the postsynaptic neuron here receives that information, okay? And so this presynaptic neuron originally received information that kind of started to depolarize its soma. Whoa, sorry. All right, so when we're looking at this cell, there would be EPSPs, all right, coming into the cell body of this cell. All right, so bringing it slowly, we'll just put little EPSPs here, all right? They're going to be transmitted, all right, and we're going to talk about that also a little later, all right, in through those dendrites to the cell body. Okay, and we'll do one more here, EPSP. Okay, so little tiny increase in charge in the membrane, all right, and so all those increases in charge are going to travel through that soma towards our axon hillock, okay? So remember, this area here, right, kind of that triangle-shaped area before the first myelin bead, all right, that's our axon hillock, okay? So all of those EPSPs are going to kind of add together and move towards that axon hillock, okay? And so that axon hillock is special. Remember, it determines if an action potential is sent or not because it has these voltage-gated sodium channels, okay? So this is a V for voltage-gated sodium channels. The rest of the cell body does not have those sodium channels. Okay, so once this increase in charge from our EPSPs travel through the soma, all right, reach this area of a dense collection of these voltage-gated sodium channels, all right, they can raise that membrane potential to threshold. Okay, so our EPSPs are going to add together. They're going to travel to this axon hillock. They're going to bring that axon hillock up to threshold. Threshold will open our sodium channels and sodium can rush in. Okay, so sodium rushes in, that depolarizes our membrane. All right, so now we're negative on the outside, positive on the inside. And then our voltage-gated potassium channels, right, are going to open at that positive depolarized potential and allow sodium to rush out of the cell. Okay, that will repolarize. All right, so what we've done is these EPSPs added together, they brought that axon hillock, right, to threshold and started an action potential at that axon hillock. All right, so that action potential, that change in charge, right, from the sodium, what's going to happen is that sodium, right, is going to move into the cell like this, and it kind of diffuses down, right, still kind of moving down a concentration gradient. It diffuses down through this myelin bead. Okay, so we have just a reminder of kind of our structure, right? These blue kind of boxes, right? That is our myelin sheath. All right, each one is going to be a little myelin bead. There are spaces in between them. Those are called nodes of Ranvier. Okay, and remember, our saltatory conduction, our action potential is going to jump from one node, right, to the next. Okay, and so that's what we're talking about now. All right, so our sodium comes in, depolarizes the membrane. As it does that, it kind of diffuses through this myelin area. All right, it's important to know that where there is myelin sheath, there are no voltage-gated sodium channels. All right, let's make a note of that. Where there is myelin, there are no voltage-gated channels. Okay. All right. So what happens is this sodium kind of diffuses across this area where there's no voltage-gated channels, and all it needs to do is bring this sodium channel in the node of Ranvier close enough to threshold that you start an action potential. And that's exactly what happens. All right, so we don't need a ton of sodium. We just need enough sodium to kind of move through that myelinated area to open this next voltage-gated sodium channel. So that one's going to open, sodium will rush in, depolarize the cell, then potassium channels open, so potassium rushes out, we repolarize the cell. Okay, and so at each node, you would have little sodium-potassium pumps working to reestablish that gradient, right, of sodium and potassium as those channels are open. All right, and so then the same thing would happen. The sodium from here would move down the myelin, depolarize it, right, just enough to open this sodium channel. That sodium will rush in, depolarize, right? You get potassium rushing out to repolarize. That's our action potential. And it would keep going all the way down the axon of this neuron, right? And so that is referred to as propagation. All right, so this is just movement of that action potential down the axon. All right, so you're just propagating it down the axon. Propagation could be used when we're talking about continuous conduction or saltatory conduction, right? It's just we're moving it down the axon, right? It's moving forward. Action potentials do not move backwards in neurons, right? They always move forwards. Okay, so finally, that action potential, right, it's going to go jumping from node to node, right, because it can only occur at the nodes because only the nodes have voltage-gated channels. Okay, so we have action potential here, here, and then you'll have another one here, and that action potential will eventually reach our axon terminals. All right, so I'm going to zoom in on the axon terminals a little because we've got some kind of smaller stuff going on. Okay. All right, so in our axon terminals, all right, we have, and this is going to be very familiar from our neuromuscular junction, right? We have voltage-gated calcium channels. Okay, and so when that action potential travels all the way down the axon and reaches these axon terminals, these little bulbs, all right, they're going to open these voltage-gated calcium channels. And calcium is going to rush into that axon terminal. Remember, calcium rushing into the axon terminal causes our vesicles containing neurotransmitter, right? So this is a vesicle of neurotransmitter. All right, that calcium moving in causes that vesicle to be exocytosed. Okay, so this vesicle will move to the edge of the axon terminal, release the neurotransmitter, and that neurotransmitter can now diffuse, all right, all throughout the space, right? This neurotransmitter, it'll diffuse, okay, and eventually it'll find a receptor to land on, all right? So the last thing to point out with this picture, and that'll lead us into our next topic about neurotransmitters, is that there are kind of two types of receptors that these neurotransmitters can find. And so it depends on the neurotransmitter as to what kind of receptor it has, all right? So these are, we'll just label them in general right now. All right, neurotransmitter receptors. Okay, and so they can either, right, you bind your neurotransmitter to one receptor here. This one causes a sodium channel to open. Okay, so this one would depolarize the cell. All right, this would cause an EPSP. All right, because you're letting sodium into the soma of that neuron, right? That's going to make it a little more positive. Remember, this is our soma, so we don't have any sodium channels, any voltage-gated sodium channels until we reach our axon hillock. All right, so we're just going to be making little EPSPs, all right, that can add together to bring us to threshold eventually. Okay, so a neurotransmitter that opens a sodium channel is going to create EPSPs. However, there are also neurotransmitter receptors that open channels for chloride. And chloride is a negative ion, all right? So that would bring the membrane further away from threshold, all right? So this would create an IPSP, okay? So you can have neurotransmitters that create IPSPs, and you can have neurotransmitters that create EPSPs. It depends on the neurotransmitter and its receptor as to which it's going to do. Okay? All right. And so then you would continue this process, right? So now, this neuron is receiving information, all right? It's going to add together the EPSPs or the IPSPs, right? And if that brings it to threshold at the axon hillock, it'll send an action potential and go through the process we just talked about. Okay? All right. So this is our kind of overview of neuron physiology, all right, how it works. I would honestly recommend watching it maybe a couple times till you have the process down and you understand what the steps are, right? Maybe write out the different steps and study those. because this is a little complicated, all right? It shouldn't be too bad, right? Because we had a couple parts of it already with our muscle chapter, okay? All right, so now let's talk about neurotransmitters. Okay, so with neurotransmitters, as I just mentioned, right, they can be either excitatory, all right, meaning that they allow positive ions like sodium or sometimes calcium to enter, and those would create EPSPs. Okay, they could also be inhibitory neurotransmitters, letting negative ions like chloride in. Those would create IPSPs. Okay, so you can have excitatory neurotransmitters, inhibitory neurotransmitters. You can have neurotransmitters that are excitatory and inhibitory, which just means that they can bind to receptors, right? Different types of receptors that could either let in sodium or chloride. Okay. All right. And we'll talk about some examples of neurotransmitters in a second. All right. And so with neurons, all right, and how they're connected to each other, they can be connected in a few different ways. Okay. And so one thing is that we, like I said, with the EPSPs, you end up kind of adding them all together. All right. And what I mean by that is it's called summation. Okay. We had summation with muscle cells. This is kind of similar to that. All right. You have two types of summation with a neuron though. So you can have temporal summation. And so this would be more like what we saw with our muscle cell. So this would be where one neuron sends many action potentials, right, neurotransmitter releases back to back, right? So this would be one neuron is creating many EPSPs. Okay, and those EPSPs would all add up together, all right, to hopefully bring that axon hillock to threshold. Okay, so that's temporal summation. And then you can have spatial summation. So this means that you could have many neurons that are presynaptic, all sending information onto one postsynaptic neuron. Okay, so you could say each neuron creates an EPSP. All right, and all those from the different many neurons are added together, right? So you still get many EPSPs, they're just each coming from a different neuron, versus temporal, you're getting many EPSPs, but they're only coming from one neuron, okay? So spatial summation would look kind of like what we were looking at here, right? So this postsynaptic neuron is receiving information from 3 presynaptic neurons, all right? So that could be spatial summation. Okay. All right. And then neurons arrange in circuits. So circuits can be converging, meaning that you have multiple neurons all feeding into one. All right. So we would have many neurons, all right, talking to one neuron. Okay, or you can have kind of the opposite. We have a diverging circuit, right? So you have one neuron talking to many. Okay. All right, and so that's what we have here. So we have this one neuron is talking to these guys, right? These guys would then talk to some other neurons, and that signal gets spread farther and farther and farther. Okay. All right, and then lastly for this chapter, we're going to talk about a couple types of neurotransmitters. Okay, and so your notes for this chapter lay out some other ones as well, all right, that you should be familiar with, but these are some of the main ones, okay? So again, remember, they can be inhibitory, excitatory, or both. It all depends on the receptors, all right? And so acetylcholine we're already familiar with, okay? So we saw it at the neuromuscular junction as an excitatory neurotransmitter. We're also going to see it in a later chapter. It's involved in the autonomic nervous system as well, right? Not just muscle contraction. So the autonomic controlling like smooth muscle and cardiac muscle and stuff like that. Okay, and then another really important neurotransmitter is GABA. So GABA is the primary inhibitory neurotransmitter, all right, in the body. All right, it's the most common. So this is going to create IPSPs. All right, so it's going to inhibit, take that neuron, the postsynaptic neuron, further away from threshold. And it's kind of opposite. The most abundant excitatory neurotransmitter is glutamate. All right, so glutamate is the most common excitatory neurotransmitter, and it is going to create EPSPs. Okay. All right, and so your chapter notes for this chapter also talk about things like noradrenaline, dopamine, serotonin, nitrous oxide, which are also all neurotransmitters and just have some slightly different functions. So familiarize yourself really just to the extent that's mentioned in your chapter review notes. All right, you don't need to go into detail because a lot of them, like dopamine and serotonin, have many very widely varying and still being studied functions. All right, so just know to the extent of what your notes mention, okay? All right, so that's it for the nervous system chapter. All right, after this, we will move on to the brain.